Let's talk about some of those differences that we see between eukaryotic and prokaryotic gene expression. So eukaryotes have more problems with transcription than prokaryotic organisms do. They can't, and the reason for this is eukaryotic organisms have a lot of DNA that doesn't code for protein. And so within any specific gene for a specific protein, there might be regions that are not translatable. And those regions we call introns, and we have to get rid of those regions first, okay? So eukaryotic transcription has to be processed before translation can occur. Hello, there we go. So there are three basic things that have to occur. We're going to first remove introns. Introns are those non-coding regions of the DNA that are transcribed initially in the messenger RNA. So we get rid of the introns, cut them out, and splice together the exons, okay? Exons, I always think of, are the expressed portions. So exon. And introns are regions that don't code. So if you're watching a Lifetime movie, this here would be like a commercial, whereas this is the actual movie, right? So you want to watch this part, but you don't care about that part as much. Once that happens, we're going to add a 5' guanine cap, and we're going to add a 3' poly A tail. So the guanine cap and the poly A tail protect the messenger RNA from being degraded too quickly, and also keep the ribosome from falling off too suddenly. Well, another major difference, which I mentioned before, is that eukaryotic ribosomes are 40S and 60S. They're called 80S subunits, whereas the prokaryotes are 70S. And they have a 30 and a 50S subunit. Okay, so remember that. That's one difference. That's another difference as well. So here's just a table that talks about some of those differences. Prokaryotes don't have the guanine cap or the poly A tail. Prokaryotic messenger RNA does not contain introns. Translation of prokaryotic messenger RNA can begin before transcription is finished. And the messenger RNA can often be polycistronic, which means it's coding for more than one protein at the same time. Eukaryotes obviously have the guanine cap, the poly A tail. They have introns, which have to be removed. Messenger RNA has to be completely transcribed and modified before translation can occur. And messenger RNA is monocistronic versus polycistronic. I'm going to quickly go over some unique changes before in this video, just because this has been a short video. So I'm going to quickly talk about some of the changes that microbes face and how they have to control gene expression associated with those changes. So microbes are constantly facing changes, really rapid changes as well. So think about E. coli in the intestinal tract. There are periods where E. coli are going to get lots of food, and there's other periods where they're not going to have any food. There's periods where E. coli are going to be in an anaerobic environment, but as soon as defecation occurs, they enter an aerobic environment. So they have to be able to turn on and turn off different switches and utilize different biosynthetic pathways depending on what's going on, what environment they're facing. Microbes use different mechanisms for for talking to each other. So they can transmit information from outside of their cell to the inside of the cell. This allows microbes to be able to sense what's going on and react to the changes. Quorum sensing allows microbes to kind of talk to each other, so they can see how dense is their population, because a very dense population might mean that they need to turn on certain genes that are useful in those more dense populations than are useful in a non-dense population. And so when we talk about these changes, you have to look at the different roles that natural selection plays in gene expression. So natural selection is the selection of certain characteristics that are more beneficial to the environment that organisms are in. And the expression of genes can randomly change. The genes that enhance survival are going to be the ones that are typically going to survive and be passed on. And so here you have, I just thought this was cute. Natural selection does not grant organisms what they need. And here you have a beaver that got a chainsaw for a hand. And you can't try to adapt. Trying doesn't work that way. It's on the genetic level, okay? One type of natural selection that allows organisms to survive is called antigenic variation. So organisms that can change their surface antigens can survive in environments that are potentially harmful to that organism. So let's think of a pathogen in our system that causes certain disease, like Neisseria gonorrhea causes gonorrhea, and it has a gene for pillin, which is a protein subunit of pili. But it can randomly move the genes in and out of the expression locus. And so the immune system may respond to the dominant pili that is there, and then they switch to a different pillin. So they're constantly changing it, so the immune system has to respond and produce new immune system cells. Another example is Trypanosoma brucei, which causes African sleeping sickness, has proteins on the surface of its cell as well that they turn on and off. And so when they turn certain proteins on, then we have different antigens on the surface that the immune system responds to. As soon as the immune system makes an army and goes to attack, they switch and turn a different antigen on. So a different surface protein on, and the immune system doesn't recognize them anymore. I'm going to stop here, and we'll talk about gene regulation in the next video. So I'll talk to you later. Bye.